Hey everyone, Dr. Jack Gordy here, and I'm continuing the journey of talking about the problems in science right now. And in this one, I'm covering translational failure. Now, translational failure is the failure of preclinical experiments in mice and cells and rats to predict what's going to happen in the clinic. So a classic example of translational failure is drugs that work in mice seem to not work in humans. At a rate, in cancer biology, the rate is around 500 treatments. For every 500 treatments that work in mice, one will work in humans, but in neuroscience research, it's way worse than that. In Alzheimer's disease, there are thousands of drugs that work in animal models and no drugs that work in Alzheimer's disease right now, in my scientific opinion, informed scientific opinion. And in stroke research, there are thousands of drugs which work in stroke, again, and maybe one that works, the clot-busting drug, maybe one that works in the clinical setting. And so the translational failure rates in neuroscience are terrible. We're talking about 5,000, 10,000 to 1 kind of success rates for what works in animal models to translate to a success in clinical settings. So let's jump into it. What I wanted to talk about is that, I covered this in my last video, there is the debate. Whose fault is it? Is it the preclinical scientists for doing poor quality research, dodgy research, biased research, or is it the clinician's fault for designing clinical trials that don't replicate the preclinical trials very well? So including way more kinds of disease, way more treatment strategies, etc., going beyond what was tested in the preclinical setting. So to explore this, I'm going to cover a case study. It's called NXY059. This is a potential treatment. Drugs are always given weird names, and this is just a name of a drug. NXY059 is kind of catchy. I like it. So let's explore what happened to this drug. What was the journey of this drug? Okay, so this drug was tested in stroke. So I'm just going to briefly introduce stroke. So there are several kinds of stroke, but I'm going to talk about the most common kind of stroke. This is about 80% of all strokes, which is ischemic stroke. Now, ischemic stroke is a clot that forms in your brain. And so now part of your brain isn't getting blood, and so it dies. And what also happens is that the part that isn't getting blood dies, but then there's a part around that part that is still getting blood, but it too dies because of all the toxic stuff going on inside that necrotic core, that area of cell death really close to the blood supply regions of that blood vessel. So that's ischemic stroke. It's essentially a heart attack for your brain. Just think about it like that. A clot in your brain that kills off a whole bunch of tissue. Now, one of our key, especially during the late 90s, early 2000s, one of our key understandings about what's happening in that potentially survivable area of the brain, it's still getting blood supply, but it's sort of getting killed by the toxic products that are leaking out of that necrotic core. One of our key understandings of that was this concept of free radicals, reactive oxygen species, or oxidants. These kind of all roughly mean the same thing, right? And what these are very reactive molecules that can be produced when things go awry in the body. But now our understanding is very different, but this is what we believed at the time. We got this massive burst of free radicals and reactive oxygen species, and these went out and damaged and killed the surrounding tissue. This is an example of how this can happen. So here we have the electron transport chain, which is inside of mitochondria. We should all be familiar with it. An electron jumps along, and that's how we generate much of our ATP, using the electron declining in energy. But when things go wrong in your body and cells are put under stress, electrons can essentially fly off this electron transport chain and end up jumping onto oxygen, and this creates a very reactive oxygen molecule. This called superoxide. This very reactive oxygen molecule can then be converted to hydrogen peroxide, which would you believe is less reactive than this very reactive superoxide molecule. And so now you've got superoxide and hydrogen peroxide in your brain. Now we use hydrogen peroxide to bleach our hair from black to blonde. So you can imagine hydrogen peroxide in your brain is not a great thing to have in your brain, right? This is a free radical. It's a reactive oxygen species. And so the idea, it's an oxidant. The idea is that we need to stop these very reactive toxic molecules from killing more brain tissue, right? So that was our understanding of the pathophysiology of stroke. You've got hydrogen peroxide in your brain. That is not a good thing. That is killing more brain cells. So they wanted to do NXY. Well, what is NXY? It is an antioxidant, right? So it is supposed to react with hydrogen peroxide and superoxide and neutralize their damaging effects, right? It is an antioxidant to prevent the oxidants and free radicals from causing damage in the brain following stroke. Seems, on paper, like a really, really good idea. So NXY is a very potent antioxidant, and it was found to be therapeutic in a number of animal models of stroke, right? This kind of experiment had been repeated like 11 times, 11 different labs, all doing animal models of stroke, all showed that it was therapeutic to some level. Most showed that it was therapeutic to some level. So there was a very good clinical case to put this into a clinical trial. So there was 11 animal models of ischemic stroke, and NXY was found to be neuroprotective. These included rats, mice, rabbits, and marmosets. So it was across the spectrum. It really was tested very thoroughly in animal models. It's definitely going to work in the clinic, right? 100% this drug, this antioxidant, is going to work in the clinic. So then they took it to the clinic and they did a phase 2 clinical trial. This was a trial of about 1,300 people. That's a lot of people. That's an expensive trial. And it was found to be sort of mildly therapeutic. But here's one of the key things here. In a phase 2 clinical trial, you're not actually testing to see if the drug is therapeutic in a clinically relevant way. You're testing whether it will alter what you think it will alter. Does it alter the amount of oxidants, for example? And what they found was there was a hint of a signal anyway that it was therapeutic. What often happens in a phase 2 clinical trial, you design it to test a proxy of a clinical outcome. So does an antioxidant, for example, stop the production of oxidants? That's what you'd hope. So you'd measure the oxidants, for example, oxidative damage going on. That's something you could measure, right? You don't actually measure, do the patients move better? Do they talk better? Do they have better cognitive abilities? Do the patients have a better outcome? That's not what you measure in a phase 2 clinical trial as your primary outcome. But what they often do is sneakily measure it anyway. So they do measure it as a secondary outcome, knowing that they haven't designed the study very well to take that measurement. But they're looking for a hint, right? And so they did this first trial and there was a hint. There was a hint that this drug was going to work. So they're like, yippee, let's push forward with a proper phase 3 large clinical trial. So they did a phase 3 clinical trial, 3,300 patients, the largest stroke trial ever conducted at its time. This probably, they didn't release how much it cost, but probably cost hundreds of millions of dollars to do this clinical trial. And so we never knew the cost that was released, but it was probably in the hundreds of millions, this clinical trial. And we know this because a similar stroke trial of much smaller people cost around 18 million in 1995. So this was done in the late 2000s, so things are going to be more expensive. It was done in 5 times as many people, so it's going to be more expensive. We expect this to have cost hundreds of millions of dollars, this clinical trial. And these were the results. So here we have the dots, that's the mean, and here we have the line, that's the confidence interval, 95% confidence interval of each of these measures. And here we have placebo is better, and here we have NXY is better. And these are the neurological scores for how bad the stroke was. These are just some other measures, if the drug was used with or without other treatments for stroke, and whether it was given under 4 hours or over 4 hours following stroke. The key thing here is none of the dots are massively over here, which means NXY is better, and none of the 95% confidence intervals don't cross this line. If the 95% confidence interval crosses this vertical line, then it's not statistically significant. It's essentially saying that the ratio is 1, right? And so a 1-to-1 ratio is no difference, right? So in other words, the drug significantly and substantially had no effect in clinical trial. We tested in 11 different animal models, all showing it was therapeutic, and then we went to the clinic and it failed. We had all this reason to think that it would work. It was a massive multi-million-dollar clinical trial and it massively failed. So, do animal models actually work? This is the big question. When developing a novel treatment, the goal of an animal model is of human pathology is to accurately predict whether a treatment will be therapeutic in the clinical setting. In the case of NXY, animal models completely failed. This is translational failure to accurately predict the therapeutic potential in the human condition. Now, I'm saying this as a case study, but this has been repeated thousands of times, okay? Literally thousands of times we've shown things work in the animal models and then fail in the clinical trial. So why did the animal models fail? Now, there's many answers to this. Some people say mice aren't humans, right? And that's reasonable. But the one thing I'll say is if I zoom in and look at some neurons and I show that to a neuroscientist, they wouldn't be able to tell whether those neurons were mouse or human, really. If I zoom in on a liver or I zoom in on a kidney, it's very difficult to tell whether that was a mouse or a human. We're made up of the same stuff, right? We're genetically very similar. We're made up of the same stuff. So are mice humans? I don't know, because every drug that works in humans works in mice, right? If you give caffeine to mice, they get uppity. If you give methamphetamines, they get uppity. You give marijuana, they become forgetful. I've actually done that. Marijuana, similar chemicals, cannabinoids. I've given cannabinoids to mice and they become forgetful. So everything that happens in humans happens in mice. So if it was a difference between humans and mice, I don't think that would be the case. I think that's a really weak argument. But this study tried to get to the bottom of it. Why is it that the animal models failed to predict what would happen in the clinic? So what they did was they took all the animal studies for NXY and they broke them up based on quality markers, right? Blinding and randomization. If you're not familiar with that, make sure you go back and watch our previous video in this playlist, right? And what they showed was over here on the y-axis, we have the efficacy of the drug. How good was the drug in the animal trial, right? How therapeutic was the drug in the animal model? And what they showed was that studies that randomized showed that the drug was far less therapeutic than studies that didn't randomize, sorry. So randomization seemed to reduce the therapeutic effect of the drug in the animal trial. This clearly shows that some biases are elevating the efficacy of the drug, and randomization, which can remove those biases, such as allocation bias and order bias, they can remove it, and after removing allocation and order bias, the drug is suddenly significantly less effective. Right? So this is direct evidence that these biases were making the drug look better than it was with randomization. What about, oh yeah, I've got to go into a little bit of detail here. So when blinding in animal models, there's actually two stages in which it's critical. In clinical trials, less so, but in preclinical trials, there's definitely at least two stages in which blinding is critical. One stage is that we have to induce the pathology, right? So as a scientist, I have to make an animal have the disease, right? And that process could be biased. So when we want to induce a stroke in an animal, there are several ways to do it, but one of the most common ways to do it is to actually feed a thread up through the artery in the neck and then rest it right here rest the thread at the entrance of an artery that branches out into the brain. It's called the middle cerebral artery that comes out. It's a very common way to do it. So by putting the thread into that small artery in the brain, I actually block blood flow. So now it's like we've replicated a clot. Suddenly there is no blood flow going to that region of the brain. Now we can leave the thread in permanently, or we can leave the thread in for a couple of hours, then remove it, which kind of models the clot being broken up. So we can do both of those things to model it. Critical thing here though, is there's a lot of technique to it. There's a lot of variability to it. And it's a very difficult thing to do. It takes a lot of training to be able to do this technique. And so there is definitely a chance for bias to be introduced into the induction of the pathology, right? Does the scientist induce a more rigorous, more robust stroke in the control animals and a lighter, less rigorous, robust stroke in the treatment animals? Now, how this works is maybe the thread isn't quite at the very entrance of the artery, for example. So they compared studies in which the surgeon was blinded to what treatment the animals were about to be given. And what they found was, again, the studies in which the surgeon was blinded as to what the treatment, what concealed is another way to use that term, were concealed or blinded to what treatment the animal was about to get after the surgery. When they were blinded, suddenly the efficacy of the drug decreased, right? When they weren't blinded, the efficacy of the drug increased, right? This demonstrates that there was bias going in here. Now, again, I'm saying it's probably subconscious bias. They probably weren't actively going out and biasing their results. It was probably just like, oh, yeah, that was a great stroke. I really got that. Let's put that in the control group. Yeah, I got that. That was a good enough stroke. Let's put that in the treatment group. And it was very subjective. They're not actively trying to bias it. Scientists want to make discoveries that are repeatable, that other people can do. So we don't want to intentionally bias our results, but we accidentally do. Very clear evidence that all of this efficacy was introduced through bias, right? It doesn't exist. It's not real. All of the difference between these bars is not real. So blinding of the surgeon affect the efficacy of NXY. Comorbidities were also a factor. So there's a real problem in preclinical research where we always test our results largely in young, healthy males. But people who have stroke are often high blood pressure, high cholesterol, they're elderly, they have obesity, etc., etc. And so these guys broke up the studies and they showed that when the mouse was hypertensive, so had high blood pressure, or rat more likely, when the rat was had high blood pressure, the efficacy of the drug was lower than when they were normotensive. So when they had normal blood pressure, right, suddenly the efficacy of the drug goes up. And so here we can see that, and similarly was found with the observer as well. When the observer was blinded, when you have to measure the stroke size by doing histology. So you have to section the brain and then measure the stroke size. They also found that when the person measuring the stroke volume wasn't blinded, the drug was more efficacious than when they were blinded. So this is 100% near perfect evidence, right, that bias influences preclinical research hugely. In fact, what they found was, once you added up all those biases, if you were to administer water, but you were to allow those biases, or saline, and you were to allow those biases to accumulate, you would find that the saline is 45% therapeutic. So it reduces stroke size by 45%, just by allowing the blinding, concealing, randomizing, and comorbidity problems to leach into your research. You would find something completely ineffective like saline to be 45% therapeutic in stroke, right? So the same trial was in 2002. So hopefully after this publication, clearly demonstrating bias is a real problem. Hopefully by 2009, 2 years after that publication, we'll start to see trends and improvement, away from 15% of blinding and 20% 20% blinding and 15% randomization. Hopefully we've seen improvement in those figures. And this study here, methodological quality of animal studies of neuroprotective agents, currently in phase 2, phase 3 clinical trials, they basically showed it hadn't improved. Not only that, they showed that it hadn't improved at all in the following 2 years, but they showed that currently there were drugs in clinical trial which were built on a foundation of animal models that didn't blind and didn't randomize. This is what this study showed. And now, since that was done in 2009, we can tell you that all of those clinical trial drugs failed. Right? So again, tens, hundreds of millions of dollars down the drain because of the house was built on a foundation of sand, right? The preclinical research was not done in an incredibly rigorous way, so it provided incorrect, inaccurate results for the clinicians to base their experiments off. So the questions, are blinding and randomization necessary? Absolutely. We have to blind and randomize our experiments. We have to blind the person inducing the pathology. That's sometimes called concealment. We have to blind the person evaluating the pathology, who's measuring stroke volume, for example, or measuring pore preference. There's tests where you see whether the mouse on the stroke side doesn't prefer that pore, for example. There's loads of subjective measures of clinically relevant outcomes in animal models. So blinding, who, what, where, and when. Anything you are comparing. So sham surgery. Sometimes we do a sham surgery where we don't induce a stroke, and then we have a real surgery where we do induce a stroke. So you need to blind what kind of surgery it is, whether it is drug or placebo, whether there is a genetic manipulation of the animals that you're doing your experiments on. Everything that you need. If you're making a comparison about it after your experiment, it needs to be blinded at every stage throughout the experiment. Who? The pathology inducer and the assessor of the outcome. When? That depends on the method, right? So sometimes and often it will require multiple stages of reblinding, right? So you'll need to keep reblinding all the way through the experiment. What are the best ways to blind and what to do if blinding is impossible? So here is an example of a method of blinding. One of these tubes contains your drug and one of these tubes doesn't contain your drug. It's a very simple way. This is often done. You'll get a friend to put tape over the label and record which one it is. Now there's a little bit of a problem with this, and that is that it does work and it is a reasonably good strategy. The little bit of a problem is say I've got group A and group B and I'm starting to measure the outcome. And I see, my first 2 animals in group A had a smaller stroke than my first 2 animals in group B. And now you've got 10 more animals to measure. You're going to be biased because you'll start to reveal the blinding. You'll start to unblind yourself by this. Now, there's ways around this. For example, you can reblind again. So you could do this for the surgery. So I inject A and B. So I do my surgeries, I inject A and B. Now I'm blind. I don't know while I'm doing the surgery, what the treatments I'm going to give. So that's great. I'm blinded. But then what I could do is completely randomize the animals, not even know what group they belong to, completely blind the animals and not know what group they belong to later on. So when I'm doing the assessment, I don't even know what groupings they're in. That's a good strategy. So how can we do that? Well, a really simple way is we keep track of animals using cage cards. So there's a mouse in each of these cages, and there's details on the card that tell me everything I need to know about that mice. What you can do is slip another card in front of that card and just put numbers on it and completely randomize it. Now you could do this before surgery and then again after surgery. You have to make sure you keep track, keep good records. But this is a better strategy, I think, because then you have no knowledge of even grouping. So you don't know if they belong to group A or group B. You're completely blinded. All your animals are completely mixed. So when you evaluate the outcomes, you have no idea as you're evaluating each mouse, whether it was given the placebo or the treatment. This cage card trick is a brilliant technique. You just have to work with your technicians to tell them to lift the card if they ever want to see what's under there, okay? And you have to be very vigorous and honest to not look under those cards. So you can blind the surgeon by doing this before the surgery and then reblind. Now this is really important for knockout mice and pre-treated mice, right? So that A-B system works reasonably well, but sometimes you're comparing a genetic deletion to a wild type mice. So does knocking out this gene is knocking out that gene therapeutic in stroke and compared to a wild type mice. Now the cage card will tell you whether it's knockout. We have to keep track of our genetically engineered animals very closely. And so the cage cards are going to tell us whether it was a genetic knockout or a wild type mice. So we need to cover those in blind before we do the surgery. That's critical. And it's a good idea to then blind after the surgery as well, particularly if you did different surgeries to different animals. So you want to have no grouping awareness by the time you're evaluating those animals for pathology. What are the best ways to randomize? This is a really important point. I think I covered this in a previous video, but often researchers would just say, I'd pluck a mouse out of a cage and that mouse I would give A, and then I'd pluck another mouse and that mouse I would give B. And so the plucking of the mouse out of the cage, you gently lift them. I shouldn't say plucking. You gently lift the mouse out of the cage. That process, because you're not biasing it, you're not hunting out a mouse within the cage, they would claim as randomization. That is not randomization. They have done studies that show that typically the first mouse you grab out of the cage has lower stress hormones than the last mouse you grab out of the cage. This could be a number of reasons, but one reason is a less stressed mouse is more likely to be in the open, and a more stressed mouse is more likely to be buried in the bedding or back in the corner of the cage there. And so if I just go for the first mouse that I can pick up and claim that that's a random mouse, it's not a random mouse, okay? Because it's probably less stressed than the last mouse I take out of the cage. So the correct way is to number all your mice or rats at the beginning of the experiment, and then use the ran between function in Excel or a random number generator. I use an app on my phone called True Random. And so you numbered your mice 1 through 8, and then you say, okay, who's getting drug A? And you select 3 random numbers. And here we get 4, 5, 6, which sounds not random, but it is. It is random, and you have to just trust the randomization. And then you collect you'll have another column for your placebo, for example, and that might be 1, 3, 8, and then another for drug B, and that might be the remaining numbers, okay? So you've actually got to number your mice first and then do the randomization, not just rely on plucking a mouse out of a cage. However, what do we do in this situation, right? So this is the knockout mouse here, and this is a wild type mouse. How do we blind that, right? We know which mouse belongs to which group, and this happens in lots of research. Like if you want to research obesity, you've got obese mice and not obese mice. You know which mouse is obese and which mouse is not obese. What do we do? Or what about this? Say you wanted to do the old A-B trick, but your drug was blue. This actually happened in a clinical trial. It was pretty crazy. It was an Alzheimer's clinical trial, and the drug actually turned the urine of the patients blue. And so they were like, well, what do we do? And what they decided to do was give a low dose and a high dose, and then compare the low and the high dose, and both of the patients would all have blue urine, so there'd be no placebo effect, there'd be no, and the patients could then be blinded between high dose and low dose. It gets complicated, but what it turned out was the two groups were equal, so now you don't know whether the drug works or not. It seems like the drug doesn't work. However, all the patients in that clinical trial seem to do better than the patients in any other clinical trial or any other observational study of Alzheimer's patients. So the cognitive decline in those patients was shallower than any epidemiological study or any other clinical trial, which is crazy. But they can't get funding to do another clinical trial. You've had your shot. It costs hundreds of millions of dollars. And so there's this interesting, I don't know, because it could just be it was a unique batch of Alzheimer's patients. We don't know anything about the drug. Anyway, important to say that I know it seems like a crazy thing that the color of the drug could influence your experimental design, but it massively does. It massively does. It happened in the clinic. So what do you do? What do you do? Well, that's a tough question. I think the most rigorous answer is to get outside help. We can't simply say that blinding is too hard in these cases, and we shouldn't do it, because look at this difference in blinding here. The drug was almost twice as effective when the people weren't blinded, right? Our biases are going to leach out. So where blinding is an impossible, I think the best answer is a person unaware of the hypothesis should administer the drugs and even do the surgeries. And also we should try randomize, we should randomize after the surgery, so any observer doesn't know which animal got the drug and which animal got which surgery. So you can start to do blinding immediately after it, unless it's an obese mouse versus a non-obese mouse. Again, that becomes very difficult. Another strategy, a very last minute, if you have to, strategy is you can section the brains of the mice, you can image them, and then you can send those image files off to someone to rename the file name. So you can actually, now you don't know if the mouse is obese or not, or hairless, or whether the drug was blue, because the images themselves have now been blinded after all that processing. Now that's not great, because that means you section the mice probably knowing what they were. And it means that like most of the experiment, you knew which mice were the obese mice or not, but it's better than nothing. It's better than nothing. But I think the ideal answer is you get someone outside of your scientific research groups, such as an animal technician, or there are actually private organizations that can run research experiments. So you just send them the experimental paradigm and the drug and they will do it. I think that's a better answer than us just muddling along and trying to reduce bias as much as we can. So conclusion, translational failure rate is incredibly high. Do animal models work? Oh, maybe not. Maybe animal models are currently not working, and experimental rigor is at least part of the reason. I would say it's the majority of the reason. Other people would say it could be biological differences between mice and humans, but I would say it's the experimental rigor. We're still using low-end numbers. We're still not blinding. We're still not randomizing. We're not concealing. We're doing experiments in healthy young male mice. We need to bump up our experimental rigor if we want it to replicate once we translate it to the clinical setting. Blinding can be very difficult or impossible, but this needs to be addressed. We should be able to, every time you peer review a paper, you should control F to do a search and look for random and blinding, for example. We are failing to improve the rigor of preclinical research, and we need to, as a community, really bump it up. And so absolutely, we need to improve blinding and randomization. But there's lots more we can do. So thank you very much for listening to this series about the problem with science. I will say, think about this. This is the problems when we're trying our very best to remove some of the human biases of research. So it's easy to point at this and say, haha, science is bad, right? But not science is even worse. Not science doesn't even attempt at blinding, doesn't randomize, doesn't self-criticize. So non-scientific arenas don't go through any of this rigor-inducing and self-critiquing processes, right? And so any other institution is far more susceptible to these biases than science. So if science is struggling with these biases, every other institution is massively struggling with it on an even greater scale, right? So don't take this as a science is bad kind of series. Saying science is still the best we've got, and we're trying to improve, but there is definitely a lot to improve on. Thank you very much.